Welcome to lesson 2 of module 1, Cells as the Basis of Life. In this video, we're going to be looking at technology and mainly microscopes, how they've progressed over time, but also some of the numeracy skills that are required for using a microscope in biology. Again, we're looking at the inquiry question of what distinguishes one cell from another, and we know that being able to distinguish one cell from another was made possible due to the range of technologies which we will speak about today. Some of the meta language, cytology is the study of cells. When we talk about total magnification, we're talking about the combined magnification of both the ocular lens and the objective lens, and we work that out by multiplying both numbers together. When we talk to and refer to micrometers, we're actually talking about a specific unit of measurement, which is 1 millimeter is equal to 1,000 micrometers. And some typical talk about bacteria, they range from 0.1 to 1.5 micrometers. A plant cell is between 10 and 100 micrometers. A human cell is between 80 and 60 micrometers, and a paramecium is the largest of those, which is 150 micrometers. When we're looking at cells underneath the microscope, there are a range of steps that need to go into the preparation and the mounting of these cell specimens for us to view. So we can talk about whole mounts, which is when the whole organism is placed directly onto the slide. But this is only possible for very small organisms. We can use smears, which are usually for cells that are suspended in a fluid, such as blood, or cells that have been scraped from the surface of an organism. Or we can also have sections, which are very thin slices of specimens, which have to be embedded in paraffin wax, and a special slicing instrument called a microtome is used to cut sections that are only 1 cell thick, which allow us to view them in very great detail. When we talk about drawing of cells, we're reminded that drawing should be large, simple, and only 2-dimensional, and drawn in pencil. And it is recommended that where possible you have ruled lines. Don't draw the whole diagram, and don't draw within a circle, even though what we're seeing is through a circular lens. Don't sketch or shade, so we just want simple, clear lines. We don't need to be artists. We want to include labels, but they want to be outside the drawing, and make sure that the label lines do not cross and that the lines don't have arrowheads. And we always need to make sure that we have a scale bar and state the magnification on which we're looking at that specimen under. So here we can see here that a few calculations of how we're going to calculate the total magnification, which we've spoken about before, but also looking at the field of view, which we're seeing underneath the microscope, and trying to work out how big the different structures are that we're actually viewing at. So we're going to do lots of practice of that in class. This is just a reference. We can see here that as the total magnification increases, the actual field of view decreases. When we talk about light microscopy, it's mainly used using visible light and a system of lenses which magnifies the images, and it allows us to view living cells in color. So that light travels through the specimen and into the lens, but the light microscopy can also include fluorescence and confocal microscopy. The cells which we view underneath a light microscope do need to be prepared and mounted on a glass slide. When we talk about fluorescence microscopy, we're actually talking about looking at cells and cellular structures or any fluorescent material that allows the light that is emitted by these fluorescing molecules to be seen against a black background. So we can actually add these dyes or these markers to the cells, and it allows scientists to visualize structures and materials inside the cells that are usually too small to view. But it also allows us to inject specific dyes and markers which allow us to detect particular proteins and diagnose specific diseases if we know which markers they will uptake and cause to fluoresce. When we talk about confocal microscopy, we're actually talking about obtaining sections of cells or tissues which are stained with markers, and these produce high resolution images of thin sections of a specimen. And by doing so, a laser light is passed through the lens to provide highly focused light, and it also allows us to create 3D images with the use of software. But the development of this technology also requires us to have this expensive software, and despite being time consuming, it does allow us to view a lot more of the specimen. We then move on to the electron microscope, which uses an electron beam rather than light to view objects, which does increase the detail of structures which can be seen. The images which are produced are a higher resolution, and we can get a greater depth of field than from a light microscope. However, it does only produce black and white images which can be colored later using different software. There are two types of electron microscopy. So we have the transmission electron microscope, which allows the electron beam to travel through an ultra-thin section of a specimen. So we can see here 100 nanometers, which is much smaller than a micrometer. But to do so, we need to have the specimen chemically fixed and onto a structure which prevents it from collapsing when the electron beam is passed through it. And so obviously that specimen has to be dead rather than living, which can be done with a light microscope. And then the other is the scanning electron microscope, in which electrons bounce off a specimen, but this specimen again has to be dead because it's been coated with a thin layer of gold, which produces a high resolution picture of surface features. However, it doesn't show internal details. Autoradiography is a method of light and electron microscopy. So we can see here we developed again to identify specific organelles or the location of molecules. And the tissue in this type of microscopy is treated with a radioactive substance, which is emitted by the cell or the specimen and produces a high resolution image. And then our final technological development within microscopy is the Synchrotron, which is a large particle accelerator, which allows structures to be viewed at the atomic scale. So it's quite a large machine, and this large diameter of the machine is needed because the particles need to move really quickly, almost at the speed of light, this radiation is quite intense, and it allows light to be produced, which can allow us to see matter at the atomic scale. So we can see here that we're getting much smaller structures being seen than previously. That's lesson 2. Make sure you tune in for lesson 3.